All right.
Excuse me, Professor.
What's up? Yeah.
I just want to make sure that you got me down for attendance because
Yep, I got you. I saw you coming in. You're good.
You got me down as well, Professor, right?
I got you.
Okay.
I did. All right. Oh, did I not share my screen? Oh my gosh. Okay, I shall share my screen. So those of you guys on Zoom should be able to see my screen. Is that true?
Here we
Okay,
go.
you guys? Okay,
Yeah.
thank you. All right, you guys can see that too? All right, that's cool. Okay, so go over that quiz first, and I guess a couple reminders. One thing, let me pull this up actually real quick. So for the in-person lab schedule, you'll probably be happy about this. So this lab, there's no reason for us to come in for that. I also so sorry, I'm gonna be off camera because for some reason the camera is behind the screens. I took away one of the in-person labs and then I also modified the times just slightly by like 15 minutes. So like the first group will start at 2:15. And I have the second group starting at 3:45, okay? Just because I was thinking if we're virtual for lecture, I might have to hop in my car and get here for lab. I realized that didn't really make sense. So anyway, I'm kind of in between class and lab, okay? Did you guys all hear that on Zoom as well? You can hear me?
Okay.
It was a little faint.
Yeah.
Yeah.
So the lab schedule modified it slightly. I'll pull it up right now, actually. It's coming up. So all I did was I took away in-person lab sessions and then I just modified the time slightly by like 15 minutes, okay? So I'll just show you guys that, but you can take a look. I mean, everything is like slow motion, so. The mouse also doesn't work right either. So anyway, so if you guys take a look here, let's make it bigger. Okay, so this is lab that was in here. I just took that lab away. You guys all see that? And then, like I said, I just slightly modified the times. So, you can just look at it here. So the second session will start at 3:45. Okay, it's just by 15 minutes. All right. Bye-bye. All right, guys, so where we're at right now, right, we are in 3 here, the week of September 7th, so we're in chapter 3, right? We started kind of reviewing it a little bit on Tuesday, right? Again, all recorded lecture, okay? So imagine if I was trying to lecture today, like what the heck would I even have been able to get through? And then you guys would have heard half of it, right? That's why I make sure that the lectures are recorded. So anyway, so let's go over the chapter 1-2 quiz because that'll kind of get us a little bit of review with chapters 1 and 2. If you guys have questions, anyone has questions, ask them, and we'll move on to chapter 3, okay? So I'll pull up the PowerPoint, probably open up the homework assignment, go over those questions, and we'll sort of review chapter 3 a little bit more, okay? Does anyone? No, we're all good? Okay. All right, so let's go to that quiz first. Okay. Levels of radiation are used in PET scans to detect brain activity. So is that true? Is it not one of the, so basically I go through kind of 2 different examples, right? When we talk about like uses of radioactivity in science and medicine, right? Low levels and high levels. So is this an example of low level or high level radioactivity? Low level, right. And what's a PET? Describe a PET scan. Anyone know? Anyone wanna? Yeah. Correct. It's a scan of the brain. Good. And what? How does the radioactivity come? Brad? Yep. So you radioactively label the glucose, right? And then you inject that systemically into the patient. And because the brain, right? When you image, you're basically imaging where the glucose goes, right? And so places that have high activity, you'll see a larger signal, right? If there's lower activity, you get a lower signal. And they code it by color, right? So you can see lower or higher activity versus lower activity. All right, so good. So that was an example of any questions about that, guys? So did you guys pick true, hopefully? You should pick true, right? All right. Sharing of electrons is characteristic of what type of bond? What do you think? Choice B. Do you guys agree we should choose B?
Yes.
All right. So what's a molecule that I talk a lot about in that chapter that has polar bonds?
Water.
A molecule. Yeah, what molecule? Yeah, somebody said it here. What did you say?
Water.
Somebody said it here too. Water. A lot about water, right? And that it's a polar molecule, okay? Kind of to go a little bit further, when we say polar, right? What do we, Brad? Okay, right, so you're kind of getting, it just really means that one part of the molecule is different than the other. But you're right, in the case of water, right, you have a partial positive charge and partial negative charge on one part of the molecule. Good. Okay, so let's move on. So again, we should be familiar with these different types of bonds, right? Okay, we should know what they are. Add more if necessary, but remember that's in chapter 2. Okay, so let's move on so that we make sure we get through this. We just gave this answer away, right? So what's the answer to this one, guys?
True.
It's true. All right. Okay, so atomic number is equal to what? What's atomic number equal to?
Protons.
What about atomic mass? What's that? Protons plus neutrons. Why don't we consider electrons when we're talking about atomic mass? Their mass is negligible, correct? But what are electrons? Why do we talk about electrons, right? Why are they important in biology? Brad? Yeah, so the electrons and how many electrons a element has, right, or an atom has, dictates how it's going to bond, right? So like other question, that's all, you know, we're all kind of, then you're focusing on the electrons, okay? So the electrons are super important. They don't have a mass, essentially. So what's our answer here then? A? I believe so. All right. Okay, so level of organization that includes cells of similar structure and function is what?
Tissues.
Tissue, good. Okay, so remember, this is kind of when we remember, well, first, in that first chapter, we talk about, right, the different characteristics of life. One of them is life is organized, right? And then I went through kind of these different levels of organization. You guys remember that? So what's the smallest or lowest or smallest on that level of organization? What's the atoms, right? And when two atoms join together, we're just talking about bonding. What do they form? Molecules. Yeah, right? Cells and tissues and then organs. Okay, so cells of similar structure and function make up tissues, yeah? So I should choose B. All right. Okay, so which of the categories listed below is most inclusive? So remember terminology here in terms of inclusive is really the, right? That's going to have a whole bunch of other categories underneath it. So what'd you choose here?
I chose E. Is that right?
Domain.
Least inclusive,
Domain.
that's the most specific. Domain, yeah, so that's why I want to be careful with the terminology, right? Because I could ask it by saying least inclusive, right? Here, I'm saying most inclusive, so it's domain, right? Remember? And so how many domains are there, guys? Nope, how many domains? I'll go back to that. Once we finish the quiz, I'll pull that figure up or whatever, just to clarify. How many domains? 3. Remember, domain eukaryote, bacteria, and then what's the other one? Eukarya. Starts with an A. Yeah, archaea. 3 main domains. Eukarya will then break it up into these other smaller categories, right? Okay. Domain, they don't get broken up into smaller categories. Okay. So we'll talk about bacteria next semester as well, but a lot of what we focus on obviously is domain or eukaryotes. Okay, so which one I should choose domain here? So hopefully that's the right answer. All right, next one. It's going. Thank you. What else can we ask on that sort of, so what's the name of the branch of science that essentially categorizes living things? Is it a T? The branch of science that is responsible for categorizing, right, and setting up these domains, right, species, and all that is taxonomy. All right, it came back. Like I said, we can kind of go back and review some of that a little bit more. So a solution with a pH of 9 is what? Basic, neutral, or acidic? This is at the end of chapter.
Basic.
Basic. Okay, so remember that we should know the pH scale. What's neutral on the pH scale? 7. Anything below 7 is acidic. Okay? Yeah? So make sure you guys are familiar with that. So this is basic. That's basic. Anyone remember? There's some examples that were given. Something that's basic. You guys on Zoom, what's an example of something that's basic? I mean, if you want to think of a chemical, sodium hydroxide, right? But also like things like milk and stuff like that are basic. What do you think of when you think of something that's acidic? Say it louder. Yeah, hydrochloric acid. That's the one that probably most people kind of think of. Also, right, acid. Ooh, I'm going to trip again. Acid in your stomach is also right. Your stomach is super acidic. There's a pH of about 2 in your stomach. Okay. All right. Includes prokaryotic organisms. So we just talked about this kind of, right? The only domain that contains or includes eukaryotic organisms is, so the other 2 are prokaryotes. So it's answer C here. Do we agree? Yeah?
Yeah.
Yeah.
Yikes. All right. Human forces that destroy the tropical rainforest lead to an increase in biodiversity. Is this true or false?
False.
False.
False. What do we mean by biodiversity? So again, that's something that's in towards the end of chapter 2. Yeah. Okay, good. Who else was going to say something? So biodiversity really is, yeah, just the number of species that exist, right? And so by coral reefs, right, and the rainforest, we basically decrease biodiversity, right? Because animals lose habitats. We also may get rid of lots of plant life. It's not so hot. In the picture in the PowerPoint, right, I think shows you the coral reef changing over time, and you can see how healthy it sort of looked. I forget what it was, like in the 70s, right? Right, so who cares? Who cares if you destroy the coral reefs, right? Who cares if biodiversity decreases? How does that impact us? What do you guys think? Or even think about the rainforest. Why?
It kind of just breaks down our whole ecosystem. We kind of need their interactions to survive. So.
Okay.
Maybe climate change.
Well, climate change can kind of cause biodiversity. Yeah, but I guess you can kind of go the flip side too, right? This sort of change in biodiversity can also sort of lead to that too. It's kind of like what about like rainforest? What kind of things do we get from the rainforest, I guess? If you think about that specifically. So, I mean, if you think about like building, but also lots of like treatments and medicines and things like that, therapeutic agents come from plants in the rainforest, right? And so if we lose all that diversity, we actually, that actually produce some of those agents, right? So that's kind of one example, but yeah, if you think about the coral reef too, we're plant life and aquatic life there, right? So anyway, just something to kind of keep in mind. But yeah, it does go along with climate change and the whole point, I guess, too, is that it's our doing, right? A lot of it is because of human activities, right, that cause that change. So we just have to be careful, right? So this would decrease biodiversity, right? Not increase biodiversity. It's going to have a negative effect. Okay, last question. On shell of an atom is called what?
Valence.
Valence. Good. So remember, that's what we talked about. This is super important. Not really what's most important of electrons in that outermost shell, right? That's what's going to dictate that atom's binding capabilities, right? And so that's why we really focus on that valence shell. In general, how many electrons are most atoms? So we'll just kind of, with the exception of hydrogen, what number are most atoms trying to get to in their valence shell in terms of the number of electrons? 8. Good. If we talk about that innermost shell, right? It's only 2 there. For the most part, we kind of think about 8, but obviously we talk about and think about hydrogen a lot, but hydrogen is only going to need 2 to fill its valence shell, okay? So again, the electrons they have, they're always going to be looking to fill it to that 8, and whether that's by, forming covalent bonds, right, or ionic bonds, that again is dictated by the shell, okay? All right. So let's make sure we got 100. If not, then I did something wrong, messed something up. We're good. Okay, so I think I told you guys this. I will change the settings so that you can always go back and look at your attempt. You probably weren't able to do that now because there was a couple people that, like, when you're studying for the exam or whatever, you can go back and look back at your quiz, right? Look at the questions and things like that. Sometimes similar questions show up on the exam, but to be kind of more simple and straightforward and really just to kind of check your knowledge, right? So I think I told you guys this too. Quiz 1 is always a wake-up call to everyone. Well, maybe not everyone. Oh, maybe I really don't know this stuff, right? Maybe I need to go back and listen to the recordings and really take notes, right? That's why I give you guys that review sheet, right? there's videos, there's anything that you possibly could need on Moodle. And again, if you have more questions, ask me, ask the group tutor, okay? It's your responsibility. If you understand something, ask. I'm more than happy to go over it, okay? But remember, you have to make sure that you know that material, okay? So use whatever resources are available. And if you're not sure, then contact me. We'll set up a meeting, okay? Don't wait until like after the first exam. Because as you can tell, even as we've moved on just to chapter 3, right? It's not gonna get less dense, it's gonna get more dense, okay? First semester of freshman year it's rough, especially if you came out of, you don't have that strong of a background. It's okay. You can get there, but just know that you're gonna have to put the time in now. And then as you move on in later courses, not that things get easier, but the courses get harder, but I feel like it gets easier for you because you understand everything more, you know what I mean? You have that basis. So if you're frustrated, reach out to the group tutor, reach out to me, reach out to your friends, whatever you need to do, okay? Don't wait, all right? So if you didn't do well on the quiz, you should be doing something about it. Good, right? But maybe even just study habits, right? Those are things that you have to kind of figure out. So that's why it takes a little bit of time to kind of figure all these things out. So like I said, I'm here if you need help. The group tutor's there. If you do need something, you let me know, okay? All right, so let's move on. I'm gonna, let me ask you guys this. So from kind of review, how do we feel? I guess particularly probably about chapter 2. Chapter 1, I think is kind of straightforward, right? Do we feel like we need more review? Are there specific questions in chapter 2 that anyone can kind of think of or that quiz sparked? Any of you guys on Zoom? No?
I'm sorry, Professor. I have no idea what you just asked. It kept cutting you off.
Me either.
You keep cutting off.
I said, does anyone have on any of the topics in chapter 2? And I'll get you, Vanessa, we'll pull that up, okay? Anything specifically that quiz sort of triggered that you're like, oh, well, maybe I didn't really go over that enough for.
I think she's asking if we have questions.
It would help if you could see my mouth, but you can't. I have questions on chapter 2. Can you guys hear that?
She's asking if we have questions like about the quiz, if we think that we need to go over anything from it, like more in depth.
Oh no, I'm good.
All right, so Vanessa just asked about the pH scale. So I'm gonna pull that up. Anything else? We have, here's chapter 2. Let me go to pH first. So there's the pH scale, right? We already talked about what's basic, what's neutral, what's acidic. Also worth noting, right, that it is logarithmic, okay? If you go from pH 4 to pH 5, it is 10 times different, not just 1, okay? So it's on a log scale. 2 steps on that scale, say from like 10 to 8, that's 100 times, okay? So it's times 10 each time. So that's what that means. If you take a look at sort of substances and their pH, so I said milk before, that milk kind of falls right just below neutral. It's not necessarily, it's basic. Egg whites, right? You know what the pH exactly of human blood is? What do you think? What do you say? It's like 7.4. Slightly basic. So what about the pH of human blood? So again, like let's kind of take a detour for a second to say like, your pH of your blood, I told you about, is 7.4. So what happens if that pH dropped to 7? Do you think it matters? It certainly does.
Yeah.
What if it just, Can you guys hear me?
Yes, you just
Yes.
a little like in and out.
Yeah, it's like long enough.
All right. What if that pH, you know, just went up like to 7.6? Do you think that matters? It certainly does. So that blood, that pH, right, has to be maintained really right at. Otherwise, it really leads to serious effects. It can lead to organ failure. What starts to happen is proteins stop functioning, right? So once that pH changes, proteins can denature. It's super, super important that that pH is maintained where it should be, okay? Yeah. So there's like metabolic, so like there's something called like metabolic acidosis. There's also other physiological problems, right, or issues that can occur that then will affect the blood pH. Yeah. Or ways that that can happen. Other questions, guys? Vanessa, you all right with this then? Yeah. Okay. So I don't expect anything like that, right? But just kind of have an idea. I mean, I guess we should be aware that if we're talking about stomach acid, I mean, acid is in the name. We know that we're down here at like pH of 1 and 2, okay? Super, super low, super acidic. If you talk about sodium hydroxide, right, that's all the way the most sort of big, okay? Hydrochloric acid on the other end. All right, let me just see, there's kind of, oh, one other thing to mention here, I think this is that human blood needs to be maintained at 7.4, right? There is a very extensive and elaborate buffer system within your body that maintains that pH. What's a buffer? So I just told you kind of why it's important in our body, but what does a buffer just in general do? Brad?
It's a type of substance that can alter the pH.
Well, it actually maintains the pH. So right, so like if you have a buffer, say that like, you know, of 7 or whatever, it's gonna make 7. And even if you add like an acidic substance to it, that pH will kind of stay around 7. We usually do a lab where did I do that? No, I don't think I recorded. You use like antacids, right? They're essentially act as a buffer. And if you add hydrochloric acid to it, it will take a long time for that, okay? So and inside your cells, that cytoplasm is buffered as well because it's important that that pH is maintained, you know, around 7-ish, okay, 7.4. So yeah, so you guys see this? So this is what I was just mentioning, right? If your pH drops below 7, it's acidosis, and it can be caused issues, also respiratory. There's something called respiratory acidosis that can cause this as well. So if you have too much carbon dioxide in the blood, it alters the pH. Another example. Okay. All right, anything else in chapter 2, guys? Yeah. Just, what was it? Just kind of, you mean just kind of like in general about that? Okay, all right, I'll pull that back up and let me. So what is this showing you here, since I have this up here? So this has to do with properties of water, right? So why is this important? What's this this pond in the winter? And somebody on Zoom. Why is this picture in our biology book? Or what property of water is this illustrating? Somebody else besides Brad.
That basically, if the water was to like overflow at the top, the life at the bottom would be able to succeed.
Yeah, so it's floating at the top, the ice. Why does ice float at the top of liquid water? So ice, right? And there's liquid water on the bottom. How come the ice isn't underneath the liquid water?
Temperature difference?
Because ice is solid.
Less
Density.
correct. Temperature difference, obviously it's warmer, right, where it's liquid. But I guess my question is, why is that ice floating on the top and not underneath that liquid water?
Because it's the first layer.
Right, because it's less dense though. That's the property of water that we're talking about, right? So if ice was water, lakes or ponds would freeze from the bottom up. You guys agree? And what would be the issue with that? Yeah. So it's just, it would just fill up and push to the top. But what actually happens here, right? That ice that forms on the top actually insulates the water underneath. So it actually makes the water underneath warmer, right? And helps keep it sort of insulate and keep that life, that sea life or that pond life alive, right? Okay? So is this property of water in terms of density, right? That ice is less dense than liquid water, okay? Again, you guys got, if like this is the first time you're hearing me say this, it's because you didn't listen to the lecture, right? You have to listen to the lecture. Like I said, if I tried to lecture live, it would be a hot because you couldn't hear me. You guys barely can hear me because I have a mask on. It would, so it's really, you have to listen to the recorded lectures. I can't stress it enough. Like I said, if you're still not understanding it, that's fine. That's totally a different story. We'll go over it, but we got to at least make sure we're exposed to it, you know, once or twice first. All right. So if you go back, guys, the water in here too, right? That we talk about, and it's important because water is ridiculously important to life, right? So we want to really understand the different properties of an important molecule. Okay. So make sure you guys review that a little bit more. Let me just see. Does anyone have any questions sort of pertaining to water that sort of came up? So I'll show you this one here. So what's this showing you? I just kind of flipped to the other slide and it probably might have given you a clue. What two properties of water is this illustrating? Oh, it doesn't say anything. Brad. Okay, so yes, so the hydrogen bonding is basically responsible kind of for these properties. That's true. Hydrogen bonding. So why does water form hydrogen bonds? Yeah. Correct. Because of that polar nature of water, right? And that you create that, so let's go back to this, right? You create this where oxygen charge and the two hydrogens, right, are partially positive. So they will be attracted to anything of the opposite charge, right? That's why water is such a good solvent, okay? Because if you put salt in water, you mix it up, it dissolves, right? Because of this. Basically, the water molecules surround the sodium and the chloride ions, and that's how it is. But even if you got rid of the sodium and the chloride ions here, water itself, the water molecules themselves stick to each other, right? Why? Because, or partially, it's not really negative, right? This partially negative oxygen here will be drawn to these partially positive hydrogens, okay? So, to what I asked you before, right? Cohesion. Water molecules cling to each other, right? We call that cohesion, and it's due to that hydrogen. They also, like Brad said, can cling to other things, right? I kind of showed you that with the sodium ions, but even other surfaces, right? Again, because of that sort of converge attraction, okay? Does that make sense? So the two properties of water that are being shown here are adhesion and cohesion. Remember, okay, cohesion is water molecules sticking to each other. Adhesion is the ability of water molecules to stick to other surfaces, okay? The movement of water from the roots, right? How do trees get water? They get it from the ground, from the roots, right? Gets transported all the way up to the leaves, right? Okay? But the way that happens really is just based on adhesion and cohesion. There's not like some like giant like vacuum that's like sucking the water up on the chemical nature of water and those two properties, okay? All right, I gotta go back to your question, right, about valence. Any other questions on this, guys? All right, let's go back here. So again, these were all these, clear with them, okay? You should know what a hydrogen bond is. Remember we talked about polar covalent bonds, right? All right. What's the difference between a non-polar covalent bond and a polar covalent bond? Covalent bond, Brad.
The electrons aren't shared. Oh, they're just like one of them is literally gone away.
Yeah, they're shared equally, right? And so if you look at examples, right, I kind of showed you this, I think, quickly here, of covalently bonded molecules, those electrons are shared equally. And therefore, those molecules are not polar, right? The bonds are not polar, okay? When you have a polar covalent bond, they're shared unequally. And that's what creates that partial negative and partial positive charge, okay? It's an unequal sharing of electrons. And again, shell. So let's now go back where it's like a backwards lecture. Sorry. Let's go back to just talk a little bit more about the valence electrons. Because that, yeah? Like I said, I don't even know. I know your face probably better from Zoom, right, than in person. So tell me what exactly kind of is your question with the valence electrons? No, like in the periodic table. Nothing about electrons. Well, that's not true. The way it's organized. Well, the electrons don't have anything to do with the mass, right? But they have everything to do with how they. The periodic table is organized in a certain way that it sort of tells you about that. That's for chemistry, but yeah, go ahead. It's organized in something existing, right? Yeah. So I think we have like nowhere on the exam will there be periodic table. You guys do that in chemistry, so I just kind of like leave that out. But yeah, in a symbol, atomic symbol, right, or on the periodic. Sodium ion and a chloride ion? Yes. It has to do with, right, losing an electron or gaining an extra electron. That's what then that's how you're gonna get a charged molecule. Yeah. Anything that I don't think it's necessary for us to go over that in here. If you want to talk about it a little bit more, right after class, we'll talk about it a little bit more. I also feel like I'm trying to get your question, but I'm on the wrong track. Okay, so let's hold that until we're done with class. Okay, and we'll talk more about it. All right. All right. Anyone else have? All right. Let me just kind of. Let me just see if there is here. So the one is that we don't talk that a lot, okay, about ions and ionic bonding. We mention it, right? But because we're mostly concerned with organic, those aren't ions, right? And those don't form ionic bonds. And so that's why we don't really kind of talk about charged molecules that much. Now, obviously, they're super important. So I in this chapter. They're super important when we talk about like physiology, right? I mean, in terms of ions, right, in your body, what's one that is, say, of a nerve cell, of a neuron? Anyone know? An ion, I just kind of showed it. Sodium, right? Sodium, you hear about like electrolytes, super important to biology. But I guess kind of right now, because we're kind of focusing on like just talking about the different molecules and kind of set it up for the biomolecules, that's okay, in terms of the kind of chemistry behind it. But we can talk more about it. But yeah, but I don't want to leave you with saying like, yeah, ions are important to biology, super important. So sodium and potassium responsible for generating an action potential, right? What's an action potential? Anyone know? Yeah.
It kind of relates to motion, so it's a synapse result in your brain. It changes the flow charge of the biology.
Yep.
And it leads to you moving and all that.
Yeah. So when we talk about an action potential, right, or what's going to trigger contraction of the muscle, right? So neuron signaling. So I always give the same example all the time. If you think about you want to like move your foot or move your leg, right? That starts somewhere here, that initiation of that movement, right? The signal being an action potential, a really long axon, and then eventually reaches that target muscle, right? And what enables that all to happen is movement of ions. Okay, so anyway, super important. That's beyond the scope of this, but you'll learn about that later. But for right now, we're kind of, again, I think because we're sort of setting up for the biomolecules, that's sort of why we're leaving out any of that talk, okay? All right. Right, so the only kind of, the one thing we talk about is sodium chloride, right? And we can talk more about that. Okay, all right, so what I want to do, we only have a couple minutes left, but I at least want to hit on chapter 3, since that's now. I am going to pull this up, because I also want to show this to you guys. Y'all listen to the recorded lecture, right? So let's just go over a few of these review questions. And again, with that said, I'll still can review chapter 3 next week, but we have on track here. So these PDFs are always on Moodle, right? They say review packet, and what they are, it's from the textbook. So if you have the textbook, you don't necessarily need to pull the PDF up for summary. And then there are also practice questions, review questions, okay? The answers to those review questions are in the back of the book. If you get answers, you can let me know. You can, I think I told you guys, you can use these with the group tutor if you want. So if we go to 3.1 there, okay, a hydrophilic, can we make this bigger? Yeah. All right, look at number 1 here. A hydrophilic group is what? What would you choose for the correct answer? What do you think? Well, look at those choices there. What do you think? Can you see?
Can we guess on Zoom?
Yeah, what do you guys think? What answer?
E.
Your answer. Zoom.
E.
Say it louder. E?
E.
Correct. All of these. Would you guys have chosen E? Right? A hydrophilic group is a polar or an ionized group found at the end of fatty acids. Yeah? Opposite of a hydrophobic group. Yes, the answer is E. So what do we mean? Water loving, right? Readily dissolves in water. What about hydrophobic? What's the definition of hydrophobic? Yeah, it doesn't like water, right? So lipids are hydrophobic in general. When we talk about fatty acids, though, they have one part that's hydrophilic, right? The polar head, and the tails are hydrophobic. And that's what that's referring to there. Okay. Number 2, which of these is not a characteristic of carbon? What do you think there?
C.
I think C. Who else do you guys think C? C is sometimes ionic. No, not true. Forms 4 covalent bonds. How many electrons does carbon have in its valence shell? 4.
4.
So we form those 4 bonds. This is also why carbon is very predominant in organic molecules and can form these, because it can bind 4 different molecules essentially. Okay. All right. Where are we? Number 3. Guy out of the way. Oh my goodness. Guess maybe there needs to be a mouse pad or something here. Okay. Let's move over. I can zoom in a little bit more. Following reactions combines 2 monomers to produce a polymer. I know we reviewed this too. What is it? We're okay. We have a couple answers. Let me ask Zoom here. What do you guys think?
Dehydration.
Yeah, A, dehydration.
Yeah.
Yikes. Dehydration. Okay. Remember we talked about dehydration reactions and hydrolysis reactions. Water is removed. A dehydration reaction occurs when 2 monomers join together. You guys remember that? I mentioned it like the chapters, maybe not 50, but a bunch of times as we go through. So somebody give, I guess it asked it in the next question anyway. All right, what's phosphorylation? I guess maybe I talk about it a little bit in chapter 3. What do we mean by phosphorylation? Yeah, so it involves that, right? Because you're talking, so it's going from ADP to ATP, you're adding a phosphate group, yeah.
So what's losing the phosphate?
Dephosphorylation, yeah. Yeah, okay? All right, so have, anything really to do with the formation of those biomolecules. So remember, the dehydration reaction and the hydrolysis reaction, we talked about when we were talking about forming biomolecules, and then also we talked about hydrolysis when we break them down. So somebody tell me a biomolecule. There's one already listed right there. Carbohydrates, what's another one? You know it. Starch is a type of carbohydrate, right? Okay.
Proteins.
Could you repeat your question?
Another type of biomolecule. We said carbohydrates. What's another biomolecule?
Proteins.
Proteins. Yikes. All right, what else? Nucleic acids. Good. One more. Lipids.
Lipids.
All right. Lipids, right? Proteins, carbohydrates, nucleic acids. Yeah? All right. Number 4 there. The monomers are the what? Monomers. What's glucose considered? A monosaccharide, a disaccharide, or a poly?
A disaccharide.
Glucose, what's glucose?
A monosaccharide.
A monosaccharide, 1, right? So glucose is a subunit for carbohydrates. Remember, like, correct, poly's more than 1. So like when you think about like starch and glycogen, they're made up of glucose, right? They're storage molecules for glucose. Lots and lots of glucose it's a big, huge molecule. A smaller molecule, one of the monomers. All right, so the answer is C. All right, we can do one more. Which of the following polysaccharide molecule in plants? Somebody said it before.
C.
C. Starch. Correct. Starch. Okay. Animals, which molecule there is energy storage in animals?
Glycogen.
Hey, glycogen.
Yeah.
Which one is carbohydrate that's listed there? Good. Cellulose and chitin, both of them are structural. All right. Okay, guys, if you please do. If you are in lab today, just stay where you're at. If you guys are in lab, stay where you're at. Because I'll just introduce the lab right now. We're not going to go on and off again. God only knows what we'll go back on. All right, you got me? You guys on Zoom? Anyone in lab today? Just stay on for 10 minutes.
I have a question though for lab.
Yeah, one second. I got you. Anyone who's not in lab today, you already heard me introduce it on Tuesday. You guys are free to go. All right, and I'll see you guys. Remember, in-person labs start next week, okay? I'll remind you, don't worry. All right, guys. Bye, guys.
Professor, I do have a question.
You too. Go ahead.
I'm still not cleared to return to campus.
1 second, guys on Zoom, 1 second. Yeah?
Go to the links at the bottom of the Zoom. And that's the
For what? For the metric?
and the microscope.
Is there 2 links? Or yeah, there is. I don't know why. But yes, yeah. All right, go ahead. Question on Zoom. Who had a question?
Me, for the lab, the thing not popping up. Like I see you doing the video before the worksheet, it's not popping up. So I just wanted to let you know before you like.
Good, then that's
For lab, we gotta do the last
an
worksheet.
issue with your computer then, because it pops up on my end.
What's
There's
the
definitely not anything wrong with it.
There's a whole
What?
bunch of pages.
Jeff,
Which is the
I
assignment?
couldn't hear you.
There's a whole bunch of pages in the assignment you put up. This is the
Yeah,
last page.
that's why I'm going to introduce it to you guys to tell you what you need to do, okay? Right now. So hang out for a second. You guys are in today's lab? Normally? Okay, all right. So I'm going to pull that up right now, okay, guys?
Before I leave,
One
Professor,
second.
I'm still
Yeah.
not clear.
What'd you say?
Before I go, I just want to let you know that I'm still not cleared to come to campus.
Hopefully if you are by next Thursday, you can come to lab on Thursday. But if not, just let me know. We'll figure it out. You have to log in via Zoom then.
Okay, no problem. Thank you.
Ms. Kaminsky,
Yeah.
I won't be able to go to lab today, but I will be assisting in
There's no lab today. I'm just introducing the lab. So just stay on for 10 minutes because I'm going to explain what you need to do.
all
But
right.
Lab starts next week. Yeah.
Oh, thank God I didn't go.
Yeah. That's why the schedule, remember the schedule really says the dates on the schedule. Take a look at that, okay? All right, guys. So
All right.
what you need to do, you guys see this up here, right? So for lab for this week, which needs to be completed before you week, whether you're actually coming or you're Zooming in or whatever, you're going to need to complete this. So it's really simple because there's a video that explains it all, okay? So this is what you'll need to complete, okay? Let me show you just the video first, just so you know what I'm talking about. It is a demo of me doing it. Exactly what you need to do in terms of the assignment. So you click it and you watch it and have your PDF.
Hi, everyone. This is going to be our
Cut that off.
demo for
The camera quality gets a little bit better as you go, so I'll apologize. Right, this was me in the summer when I couldn't get into lab. So I explained everything there. So you will need, I'll show you this PDF here, okay? So you're gonna need to have that PDF with you while you're listening to the lab, and you're gonna be completing it as I go along, okay? That is due before you come to lab next week, because next week you're just doing some parts of it, right? Like for you, that basic, and then a paper that says, you know, weigh this, then do this conversion, or put this slide under the microscope, you know, and I'll be there to help you guys or whatever. Recomplete this whole lab, okay? So it allows us to take a shorter amount of time live in the lab without having to go through all of this. That's why we're doing the video ahead of time. it's up, we don't have our full lab time. Okay, you guys hear that? So you have to have this with you. There is more than one page. If you download it, you'll see there's two things. If you like, you can print this out and handwrite it in. That's okay, okay? But, I mean, that's kind of wasting paper, right? Something like Kami, right? Or Adobe Pro or something like that. And I think there's another way of doing it where you can just write in the PDF, okay? That's the best way of doing it. Yeah. I feel like that's like the most useful, and it's good with like Google Docs, Kami. It'll automatically put it right there. Kami, see that? Doing is just to use that. Go ahead. Who has a question?
So we have to do all the pages on that.
Correct. I explain it in the video. So some things are omitted, but you won't know that until you listen to the video or watch the video. Okay? So you click on that link, that first one, part 1, have your PDF with you, and then you're going to submit it through the assignment, okay? So the assignment is here, okay? So once you can follow along, you listen to it, and you complete it as you go. Everything's explained there.
Do we need a microscope in our hands?
Nope, that's why you're going to come live next week to actually use a. It's just learning and getting the background information. You're just watching me operate it. But that's why it's important to come to the live lab so that you actually know how to use them. Do it, right? Okay, so that's it, guys. I wanted to make sure I introduced that. That is due before you come to lab in person. So if you're in Thursday lab, you guys, the due date might say Tuesday, but you guys have until Thursday as long as it's before you come to lab, okay? Yep, submit it online through the, you don't need to bring it with you or anything like that to lab if that's what you're asking. Okay, guys, you don't need to have that with you when you come live, but it needs to be submitted via Moodle before you come. All right, if you guys got it, you're free to go. Same thing here. If you guys got it, you're free to go.
All right, that's the only thing we have, right?
Excuse me?
That's the only thing we have for homework?
Start working on that. Yeah, you might as well start working on it now if you want.
Yeah, I am. I'm just saying.
All right. Yep, that's it. You can go if you want. Okay?
I have a question, Professor. So for Tuesday, I have lab on Tuesday, but we have the lecture. We're doing the lecture virtually, correct? And then if we have lab in person, we go lab in person?
Remember, I modified the time slightly. So if you're in the earlier time, it'll start at 2:15, okay? The later time will start at 3:45. But yes, if something changes, I may say if you guys want to come in person, you can. But for now, yes, virtual for lecture, come live for lab.
All right, thank you.
Yep, you got it.
Professor, I had a question.
Sure.
I wanted to know, like, since you modified the time, is it still, like, you know, for the people that have...
Yeah, yours is not... I only modified it by like 15 minutes anyway. Yours isn't changed. You're the same.
Okay, thank you so much.
Take care.
Thank you. Bye-bye.
I have a... Let me stop the share.